Good evening and welcome to Season 3, Episode 4 of the Late Night Conference. My name is Wilhelm Huck and I'm your host tonight. And it's a real pleasure to see so many of you this evening live in the audience at Radboud University Huygens building, and also welcome to everyone who is watching online. So in this third season, we are exploring chemistry and AI. And we looked in the previous episodes, we looked a little bit more into the technical details of AI and how you could apply it to molecular sciences, and especially on the area of maybe materials design. But this week, we are going to look at another topic and really sort of looking at the development of AI for drug discovery. And we can well imagine that this is a very fruitful area of future research with huge implications for actual practical applications. And I think if you see where a lot of the big tech companies are going, they have discovered AI, of course, but they also discovered chemistry, and especially the area of pharmaceutical sciences, because discovering new molecules is extremely difficult. It's very much of a hit and miss process where thousands of molecules that go into the pipeline and only one makes it as a final drug. So you can well imagine this is a field where AI could contribute a lot. And tonight's speaker is really someone who is an expert in that field and who has really made it her career to develop generative AI to discover molecules in this particular space of applications. It's my great pleasure to introduce Professor Francesca Grisoni to you. She's from the TU Eindhoven Biomedical Engineering Department, and I think also Institute for Complex Molecular Systems. I'm not going to reveal yet what Francesca's real background is. She will do herself, she promised me. But in 2016, she did a PhD in Milano, where she was already working on AI and molecular property predictions. She then thought that she should go into the pharmaceutical world, where she joined a pharmaceutical company and a startup company. But very rapidly, she decided that that would not be her career choice. So she went to the ETH in Zurich to do a postdoc in 2017 with the Schneider Group, working on generative AI, again for drug discovery, now for real, I guess. She stayed for a little while as a senior postdoc, but then in 2021, you joined the Eindhoven University, and she is also involved in our big growth fund consortium, so I'm sure we will have a chance to collaborate in the future. She has been extremely successful already. She got an ERC Starting Grant last year, Young Career Fellowship from the KNW, prizes and awards, but that's not what we're here for to celebrate. We're here to hear more from Francesca. The word is yours.
Very much. Can you hear me? I guess so. So thank you for the nice introduction. It's great to my talk will be about drug discovery and how we can accelerate it with AI and deep learning in particular. It's working. It doesn't work. Okay, now it works. Great. So yeah, the deal is that actually I'm an environmental scientist by training, and start by talking about my profession. It's the tradition of this series of talks. Actually, the reason why I studied environmental sciences in the beginning was that I could not decide which subject I liked the most among chemistry, mathematics, biology, so on and so forth. So my decision was to not make a decision for a while. And so in the end, I discovered machine learning. Really made me super passionate about it and the potential it had. So and really was to be a guest PhD. I was working in the toxicology domain. There were other people that were working in drug discovery, and it was really, really cool because you could just test your model. Predictions were working, and we could not do it really in the toxicology domain. So for me, this was like eye-opening, and then I tried to change, and slowly I steered in that direction. Of course, I also took a break in industry to see how it felt, and I decided to come back. And since then, I'm really focusing on was focusing on bioactivity prediction and then on de novo design and how we can develop new technology to accelerate the drug discovery process. So if I could people here, this would be just keep an open mind, don't try to force yourself into a linear path at the beginning immediately, but just take all the opportunities you can, because sometimes you might find something of even more than what you're doing, which in my case was drug discovery and why actually we need, or we could benefit from machine learning. Said it well, and maybe you know that humans are getting increasingly inefficient at discovering new drugs. So we are not very good at treating all diseases, and one of the reasons is that it's extremely challenging to have even just one molecule be able to pass this entire pipeline. And there are many reasons why it is so, but one of intensive and cost intensive, and some you might spend a lot of money and still have that no molecule actually passes the entire. Reasons why it is so complex really boils down to the dimensionality of our search space. So there is such a thing, the thing that we call chemical universe. And this chemical universe is that theory make and test in a lab are contains. And this chemical universe is estimated to contain molecules. So it's a huge, huge universe. And the idea of how vast it is and how dark it is for humans, we only 10 to the power of small molecule drugs in that huge, huge universe. So it means that it's basically unknown for us. And again, to realize how big it is, you can think that it contains way more molecules than there are in the Milky Way, or is to navigate a smarter way to. This is really what we want to achieve artificially. They know that there have been many good speakers already before me to tell when we speak. Seen many breakthroughs. We almost daily see AI was published or is useful. And actually these breakthroughs are in the cases driven by what we call artificial neural networks or deep learning. These methods are really inspired, even if loosely inspired, by how the human brain works. And these types of networks have led to complex games and beats at it, or even cars that can drive around without a driver. And I really like these two examples because they somehow represent what we want to achieve in drug discovery too. So basically, we want to develop algorithms that will tell us next move make, for example. So which molecule should I make next? And where should I drive in this vast chemical universe that is completely uncharted? But how do we actually do it in practice when it comes to drug discovery? We think of it as of molecular structures and a set of properties. And in our case, what we're interested in is predicting the bioactivity. So whether a molecule will or will not interact with a certain target like a protein. And basically what we want to do is some data these two spaces, structure and property. And we can use this to try and chart a chemical universe that is two directions in which we can go, but let's say the more traditional direction is to go from the left to the right in what we call Qsar, quantitative structure activity relationship. And it's simple in its functioning, and we can think of it in this way. Is actually embedded in the molecular structure itself. In our case, it's the activity, so it is. So let's say then the pre-deep learning would tackle this problem of molecular descriptors. And these molecular descriptors are nothing. Chemists or more chemists sat down and learn from. what people have thought to encode. By deep learning, we can actually bypass this step entirely. Raw representations of molecules, such as a molecular graph, which is very intuitive, as you see, where each node is an atom, and these atoms are connected by edges, which are bonds. You saw already in some of the previous talks, actually capture the 2D information into the form of a text. And actually, what changed the game is that by actually have models that are way more accurate and capture better information in a more sophisticated way. So when I joined the TUE, one of the main questions we started from was, is this really true when it comes to property prediction? Paradigm perform better than just engineering. So to answer the question, we decided to actually push machine learning at its limits, and I will tell you why I'm talking about limits. And we decided to look into what we call activity cliffs. I don't know if you've ever heard this term, but basically activity cliffs are pairs of molecules that are very, very similar in their structure, but in which a very tiny change leads to a drastic change in their properties, in our case, biological activity. So you can see why already machine structural information, right? And recommendation when it comes to activity, observe them. So say, okay, usually people consider activity cliffs as a source of disappointment, and people just escape from which we have such activity cliffs. But actually we thought, is this really the case? Are these activity cliffs something we should run away from? It's not necessarily the case, right? Of course, the interaction of a ligand with the protein is very complex, and we have many determinants of these interactions. Like we change an atom and we miss a hydrogen bond, then the property of a molecule will change drastically, right? But if we had the then this oracle would know exactly what effect change would cause in the activity. So we thought, okay, why don't we actually leverage activity cliffs to learn something more about our models? So that's what we did, and in particular, in the study led Derek, a PhD student. So he spent a lot of effort in curating data sets, determining and quantifying activity cliffs, and then he used these activity cliffs to check the performance of several machine learning and deep learning models. And the question was, does deep learning work better on these activity cliffs, and how much do these machine cliffs? And actually the answer, to our surprise, was that, this is just a summary, you can read the paper for more details, was that actually deep learning models, well, first of all models indeed struggle with activity more than machine learning, traditional machine learning approaches. So the features to perform better on these. For me, what was really interesting was not only that, yeah, indeed, activity cliffs, also that these. Oh, what happened here? Sorry. So we started to look into the data sets and to the specific cases, and what we saw was that we had very, very different performance. Performance on a set, so you just take some molecules out and see how your model. You see that basically the better a model performs, the better it performs on activity cliffs. But there were some interesting cases. But when you look into how they perform on activity cliffs, some of them are just gone. Some them in a prospective application. So cliffs are actually friends as well, because they're very, very extreme edge cases that can help you learn something about your model. What would differentiate? Consider activity cliffs. The response was that it really depends on how many molecules you have to train your model from. So basically, whenever you have very little molecules for your model to train, you might end up with these very chaotic landscapes in which models might not perform as good as you think. And this is not surprising, because machine learning needs, in general, a lot of data to learn from, and deep learning is particularly. They really set the basis for some of the ongoing works. These models in the low data domain. We still don't have final results, but I just wanted to give you a flavor of what we're doing now. Here is that we're trying to find better ways to learn from small data regimes that are typical of drug discovery. And one of the projects going now, so-called self-supervised learning. And the idea here is that although we might not have data on the problem we're interested in, we can still leverage all of the molecules that we know and that are deposited somewhere, try to use them to teach something to our model, for example, some chemistry knowledge. Another project that is ongoing with graph neural networks is so-called active learning, where the idea is that you start with a small data set and you cannot test thousands of molecules to improve it. You can still ask the model to which model, and so then you can go to the lab, test those molecules. The idea here is that if you do it in a smart way for a few iterations, you can actually improve your model faster and smarter than just. So we will bring this one. I hope it will be interesting and satisfactory, but for now, this is all about, let's say, the forward direction of this mapping. But actually, as we said, we can also go the other direction, and we would call it de novo design. And what's the idea of de novo? That the model to screen large libraries of molecules, we can actually try and have models that, given property we're interested in, for example, a molecule that is modulating a protein, and this is really cool, because this is one of the topics in which deep learning has led to the biggest change in the landscape of how we do this compared to more traditional. And actually, one of the topics and a lot of efforts in is the so-called language modeling. And you might be wondering why I'm talking about language right now. But let's first speak about what it is, and then I will tell you how and why. And basically, whenever we talk about net that aim to learn something from large corpora of texts, usually human language. And for example, we could train these models to learn in a sentence or of a given sentence in a language. And these models can also be used to generate new language, for example, with ChatGPT, in case you have been exposed to it. But how can we use it for chemistry? What elements of, if I give you this, connecting them to each other, you will end up with, right? Because syntax, right? Only certain combinations make sense. Also, when we design new models from scratch, what we're really interested in is the semantics. So we know that based connected to each other, my molecule will have certain high-level properties, physicochemical properties, biological properties, and this is the part of with processing way. Well, the solutions, and we have mentioned it already before, is to resort to strings or selfies, where basically the idea is that it's just that I convert my 2D structure into a string. And then I can do some learning on top of this type of representation. And there are many ways. Years is to just take molecules, convert them into this string. It's one at the beginning and one at the end. The signal the beginning and the end of the sequence. And then we can use these characters. But anyway, we can use these characters and feed them into to predict at each step of my sequence what character should follow. Why is it powerful? Because if I train this network long enough, it will learn at any portion in my smile strings, right? Once a network has been trained, I can use it as a generator, right? I can start again from my go character, let the model generate the next one, feed it back, let it generate, and I can just keep going until the, right? So by just learning data, the model generates strings. Then, of course, we hope these strings possess desirable properties, so they're valid, they make chemical sense, so they can be converted to a molecule. But already seen it before, is that with drug discovery, usually our data sets are very, very small. So sometimes we don't there are a few tricks. Your data set, let's say focus data set is not enough. For example, your bioactivity data set, you can try and search for a bigger and use it to give an initial training to your model. So in our case here, so we resorted to this database called Campbell, and Campbell has 300,000 molecules that were annotated about the target, but we used the smile string to pre-train our model in that way, just to predict the next character. And what we saw was that already after this first step of transfer of pre-generate new strings, these new molecules, which means that by just showing the model some strings, it can actually learn the grammar of my chemical language. Only just the grammar, right? But was the semantics. So we took this model and basically and in this, we're interested in modulating later. We asked it to generate 1,000 smile strings, which we ranked with our usual tools. And brought up. So and these are the 5 molecules he picked, which he was not expecting this to work this well, but 4 out of 5 molecules actually has the bioactivity we were hoping to see. And it's impressive if you think about it, because the model just saw smile strings all along. We never specified what we wanted, but it somehow learned it automatically. So this study was path I might hear with me along for example, was about trying to understand what's the effects of whatever we put in our fine-tuning set on the properties of designs. And in particular, here we use natural products, which are a source of inspiration for drug discovery. So we had these 2 sets. And basically, we wanted to monitor how the properties of the designs would change based on what we put in the fine. Which is the fraction of sp3 hybrid, is considered a good indicator for the chances of clinical success of a molecule. And molecules, and in white, generate molecules after every, I think, 10 epochs and check their properties. Basically, what we could see, and it was really nice, was that somehow the properties of the pre-training set, properties of our interesting, because it's interesting because we can decide where to stop in this journey between the initial set and the final set. This molecule, and here we have idea in hand. We decided to go to the wet lab again, find some bioactive molecules that were inspired to natural products, and we decided where to stop in this journey. Somehow still preserved some of the intended bioactivity. But one of the bottlenecks in our pipelines really the wet lab part. So you always need to have a chemist that goes to the lab and spends a significant amount of time and effort in making these molecules. And usually they can make only 3, 5, something like that. So we started thinking about how we can speed up this process. And back at the TTH in the group of Gisbert Schneider, what they had this platform that could perform 17 one-step. Thought, okay, maybe this is a good automation side. But then we realized immediately tools have no knowledge about the underlying spiral. Well, here we had quite a restricted, I'm sorry for the quality of the figures, don't know what happened, but here we had quite a restricted, let's say, set of. Did we make it work? Well, we realized again the properties of whatever you put in either the pre-training set or the fine-tuning set. So we realized that by pre-training with molecules that were predicted to be synthesizable in this platform, then also the designs have higher chances to be predicted as synthesizable. So we pre-train with potentially synthesizable molecules with bioactive molecules. So here you can see a bit of a overview, and basically we were quite happy to see that the model was generating more than half of the molecules that were not contained in any database. But what was interesting was to see that the model could reproduce some modulators of the intended targets, either patented or present in Campbell, that we never used for training the model. So for us, this was already quite an indication that the model could indeed. So then we picked all of the molecules and we selected them for synthesis, and a bit more than 60% microfluidics platform, which means that somehow in a data-driven way, the model was able to actually pick up something that we needed for making purification, which is quite a demand. Just if they would show. And it was quite interesting to see that almost 70% of those. So we picked, I think, the 14 most interesting ones, synthesized and then purified them again, and 12 out of 14 showed the intended biological activity. So for us, again, because I think it was the first time that so many molecules generated by them actually are very interesting. But of course, it's not easy that one could. What so one of the bear with me. So infusing some chemistry knowledge into these models. Think of is that molecules are non-directional while language is. So when you generate language, you either generate from left to right, from right to left. And what we see unidirectional generation process, so we generate in both directions, actually we could improve designs. But also we can go the other way around. From the natural language domain. So here, imagine that you have a smile strings, and what is to model to or not, and basically and we applied this concept by novel kinase inhibitors, and this also worked. So it's quite interesting. But there's still quite a lot to do. Still struggle with quite a bit is to design actually novel and structurally novel architectures from the explore that part that is of the chemical from what we know. And also the things we're currently lacking, properties of the design. So we and then we to find a way on but what if I wanted to control multiple properties at the same time, for example, for pharmacology or selectivity optimization? How in the future and can be steered in the right direction. I mostly spoke about small molecules, but actually these language models work in the alphabet. Using SMILE strings, you can use an alphabet where every letter represents one acid. And we applied this a few years ago already when I was at ETH, and it was more or less the same as I showed you with small molecules. And again, we brought this idea to the lab, and we were able to find 10 new anti-cancer peptides that were validated in vitro. And the students, Eve, and basically the idea here is to, well, imagine you have sequences of proteins there. The idea is really to use this protein language model that would catalyze specific reactions that we cannot yet catalyze with enzymes. And I hope this will lead to some cool results in the future. But it's a really, really cool time to be working in this domain. And I think we have the tip learning in. We're getting very, very and we're getting very good at predicting, at generating new structures. But do we control the properties of our designs? How do we work in small data regimes? How have 2, 3, 4 molecules involved? These are still open questions, and I hope we will see. So that's it from me. And before I'm done, there are many people I should thank because all of these results were a collaborative effort. And or medicinal and chemistry collaborators in the group of at the TUE, and then of course our
Thank you, Francesca. You can take a seat.
So sitting.
Thank you so much for a wonderful presentation, beautiful presentation. Really nice that you explained so clearly what the tools are that you use, but what struck me also is that you clearly always show the angle on how to apply it to actual molecules that have a certain function. And the function was always, of course, in the sort of peptide domain or the sort of small molecule inhibitor domain. And so it's really nice to see that direct link to discovering actually useful molecules that have a function. So that's a real nice eye-opener. And it's quite clear that there's so many more things that can be done and will be done in the near future. There is plenty of opportunity to ask questions. Also people online, if you put them in the chat, then we will pick them up and I will see them on my screen here, I hope. Oh, it's now protected. You have to deprotect it. If you have questions, then please speak up or come to the front because the speakers are not working. Oh, the microphone is working. Perfect. Then, are there any questions in the audience?
Thank you. Thank you. Do you hear me? More or
Speak
less?
up a bit.
Louder?
Louder,
Okay.
yeah.
So,
Better.
my question is related to this de novo design of a molecule. So, what do you think is more challenging, having the property that you want in that molecule, or having a molecule that doesn't have 1,000 of problems that a drug can have when you try it in an organism? So, what is easier, like positive design of a property on molecules, or predicting a molecule that it's safe and doesn't do what you don't want it to do?
Yeah, let's say that the first stage is really, and it's the challenging one, is really to find a molecule that binds to the target you're interested in. So usually this is done in steps, and once you find that, then you can start, let's say, tweaking and playing around with everything else in the hope of making it safer and absorbable, and have it distribute in the body as you want. I'm not even sure it's the right way to approach it, because ideally, if we had very good models, we could tackle all of the properties at the same time and already generate a structure there. I would not be able to tell you which one is the difficult side, but the sequence is always bioactivity first and then the rest, because bioactivity is what determines a molecule being interesting for the pipeline. Yeah.
Thank you.
Francesca, I had a question myself, because you showed in the beginning of your talk, where you were testing the different models around your activity cliffs, that it seemed to be you needed 1,000 molecules in your training set, which to me already seemed like a very small number. But then you showed later on, when you did the ChemBL database, which had 300,000 molecules to pre-train, when you were doing the fine-tuning, you only used 25 molecules. So to me, it seems such a small number to pick up any information from. What do you think is the model really sort of picking up on? Because it seems to me that it cannot really capture the full diversity of chemical space if you have 1,000 molecules, or definitely not 25.
Yeah, so I think, and this is one of the limitations, so the model doesn't pick up everything there is to know, so that's why we struggle with the diversity of the designs. What we see is that somehow the model is very good at exploring the neighborhoods of the molecules, of those 25 molecules, and it does learn some higher level features, because otherwise you would not have binding, so it does learn some three-dimensional information, although it's trained on smile strings. But yeah, when you want to really push it outside of the regions where you have that handful of molecules, that's really difficult with those numbers. So I think we would need way more information.
So there's a challenge there, because you need more experimental data in that case.
Yes, indeed. Or to use what you have in a smarter way.
Okay. You seem to have an idea in mind, I guess, or?
No, but for example, I was thinking about a few studies that have been published recently where, for example, they try to cast the problem as a translation problem, where you have a, let's say, translator model that translates between the space of proteins and the space of ligands. And so basically this means that if you can train this model properly, it was not brought to the lab, so I don't know, but ideally you can leverage all of the information that you have of pairs of proteins and their ligands. So even then, when you move to a target that has only a few ligands, actually you can use all of the other information. But I feel like this is still in its infancy, but maybe resorting to some information on the protein side, at least in our domain, might alleviate a bit the requirements for data.
So when you go back to the compounds that you picked up from your sort of small starting point fine-tuning, if you look at, can you sort of put an attention layer there, so to see what sort of features the molecule, the model is picking up on?
Yeah, so we tried it. We never published it. So basically, what these types of networks learn is a probability. So basically, you would have that for every time you try to attach a character, every character will get a certain probability to be attached there, and so you can try and look into that. And my impression, but then we never did a systematic study, was that indeed the model was able to pick up a few relevant things, like things that are anchoring points in the binding pockets, but also some very different things that you cannot, but indeed it's very cool to open the box and look what the model is doing. Yeah.
Let me check in the audience. Tima.
Thanks for your talk. I'm wondering how did you define the features or the properties for the molecules that you synthesized by the machine learning? And for example, how did you define the chemicals, the molecule that you synthesized is toxic, or they have the kind of adhesion functions like that?
I'm sorry, I'm not sure I got the question. Can you speak a bit louder, please?
Oh, yes. I'm sorry. I'm wondering, how did you define the features or properties by the machine language? And for example, the molecules that you synthesize is toxic, or they have some adhesion function that you can figure it out by the machine learning, or now it is still a problem?
So let's say for how we set it up here, especially with the experiments performed by Daniel Merck, we always had a check on cell toxicity as well, and we would just get rid of anything that seemed to be toxic. But in theory, when you look at it from a machine learning perspective, you can also, and that's what I was doing in my PhD, you can also train models to predict toxicity. So in theory, you can have a battery of models or a model that predicts a battery of assays or properties and use that to, for example, decide which designs you are not interested in and just consider something that has the full array of properties you want satisfied. Does it make sense? Okay, cool.
This is an interesting point. So you say you use a battery of models. Are they combined into one, or do you use them sequentially to just weed things out step by step?
It's really up to you. There's no
Okay,
yeah.
you can do
There's
both.
no rule. You can do everything you want.
Okay.
Yeah.
There are a couple of people online who have questions. Dr. Zara Palavanyali, I hope I pronounced that right, asked, could you please explain more about the application of ChatGPT? Thanks. I think it's more, I guess, of the large language models.
Yes. Yeah.
So I guess maybe if I translate the question is, would you sort of say we should sort of move as a field more towards using the large language models, as you seem to be doing, or maybe the graph neural networks or some other model?
Yeah, so my answer would be again that there's no like single approach to things, and what the literature seems to be suggesting is that every method is good for their own thing. And for example, generating molecules seems to be much easier when it comes when people are using these language models, while it's trickier still with the methods we have to generate molecules in the form of a graph. While, for example, with bioactivity prediction, it seems that traditional approaches with descriptors are still the best thing to pick. So I think it really depends on what you want to achieve. But definitely these language models are super powerful and makes certain tasks so easy, even predicting reactions like many people are doing, Connor Colley, Philip Schwaller, and the people at IBM. So I would say pick the right approach for the right task, and that's it.
But I guess if you use the large language models for synthesis, then you have, of course, a huge data set because of all the chemical literature ever published will tell you if a reaction worked or not. I guess if you think about the activity of a molecule, then as you were showing, the data set is much smaller. So this is also a question coming in the chat from Stan Gielen. You described many interesting applications for AI in chemistry. Do we have enough data already to address all of these topics? Or do you say there are certain areas where we really struggle with having enough data?
Well, I would say drug discovery is one where we struggle, because I mean, for how I see it, we're designing these new tools and then using them in cases where we have at least a handful of molecules. But in a normal traditional drug discovery pipeline, when you just discover new targets, then you have no data there. So that's quite tricky compared to, for example, organic synthesis or protein science, where you have a lot of data. So I would say that's one of the reasons why drug discovery is one of the most challenging applications of AI in the molecular domain, I would say. I don't know. I would have to think about something else, but nothing comes to mind.
Other areas, I'm sure predicting emulsion stability.
Oh yeah, right. Yeah. Don't get me started. Yeah.
Field or the physical chemistry side, there are definitely lots of applications. I was thinking, you mentioned earlier you have high diversity and low diversity data sets, and so I guess when you look at it, you will immediately recognize whether it's low or high diversity. But how do you know this? Is there an algorithm that tells you this is high or low, or is there a measurement of level of diversity? How do you know this?
That's a very good question. And one of the things I tell to the students when we speak about molecular diversity is that similarity is in the eye of the beholder. So basically, I guess, if 3 chemists would look at a set of molecules, they would see slightly different things in each and every one of them. But indeed, we have ways of quantifying this similarity via these descriptors. And there are many types of descriptors that people usually apply to quantify it. The well-established one is the so-called Morgan or ECFP, Morgan fingerprints or extended connectivity fingerprints, which basically just capture the presence of certain substructures, radial substructures, and use that to say whether 2 molecules are or aren't similar. But yeah, again, we have some well-established things, but then every person might want to look at it in a different way, and so choose different descriptors. But yeah, we did use extended connectivity fingerprints for the natural products study.
I have more questions on that, but let's see if there's people in the audience. Yes, over there.
Hello, yeah, thanks for the talk. I had a question about chirality, because there is a very fundamental difference in how, like, we as medicinal chemists approach things. We like flat, rigid molecules. Then in nature, you see a lot of very complex, highly chiral compounds with a lot of stereocenters. So my question is basically, do you take chirality into account when you train the system, and does it also predict chirality in the models?
So SMILE strings accept characters that represent chirality. They're special characters. And I think at the beginning, with the first study I showed on de novo design, we just ignored those because they were just making our life more complicated and were present only in a few molecules. But then progressively we started to include those. So there is a way, and if your fine-tuning set has many of these elements, then the model will somehow pick it up and start generating that as well. So yeah, we can do that. While with the activity cliff study, we just removed, we also have this, they're called chiral cliffs. So where just a change in the chirality makes the property change in a drastic way, because there's no data really. So sometimes you would not know which, so sometimes it was just easier to delete that. But with de novo design, it's easier, yeah.
Oh. Hi, thanks for the talk. So you showed a graph, it's working. You showed a graph at some point where you trained the model on different numbers of molecules. And it seemed quite nonlinear when I don't know if it's true, but when you hit the point where your model has predictive power at around 1,000 molecules, do you know why? Or is that even the case? And is there a way to quantify the information?
So I drew just a line for our eyes to see something. There's no, I mean, you can just see the trend, and it's well known that the more data you have, the more, the better the model is.
But
so
I guess the question is why around 1,000 and not 100,000?
Yeah, I don't know. I guess it's also, maybe it's not really about the model or about the number, but it's about the type of data we have in there. In the sense that what you see, I don't know how it is in other domains of chemistry, but like in medicinal chemistry, when you look at the data sets, very often you have a series of analog molecules. So then in that case, okay, you can have 1,000 of those, but there's not too much in there after a while it plateaus. So I guess, go, yes, please.
Is there a way to quantify the sort of informational content within a
No.
molecule?
We haven't done that, but it might be interesting indeed. Some form of like, information content indeed. We should try that. I will see what we can do. Thanks.
If you take that a little bit forward, do you also have ways to, say, generate new molecules that would provide you with maximum information for the sort of property that you're trying to predict?
Yeah, we're trying something similar when it comes not really to the generation part, but to the active learning. So it's more of a predictive thing. So we basically are screening a library, and we're trying to think about new ways of actually picking the molecules that would provide the maximum information to our model. So, but that's indeed a good idea. Yeah.
I'm looking around. Well, I think you know always sort of many answers to all our questions. So I thought let's ask you another question. First one on synthesis, because you showed us a bit of synthesis where you had a flow reactor that surprisingly could make actually quite a few of the molecules that you had selected. But normally your flow reactor is limited to a certain set of chemical transformations, right? So how do you see this in the future when you really need to make your design, build, test, or make, test cycle where you have to synthesize maybe very complex molecules? what is your current thinking? Is this something you think you're going to pick up yourself, or?
I'm not sure if we are going to pick it up. I'm an environmental scientist after all, so I don't think it's where my knowledge lies. But I do think that we are getting very good at learning directly from the data. So this strategy, I don't know about like the synthesis part, but actually if you find a way like we did to be able to even just roughly detect which molecules might be somewhat suitable for a given platform, then it's already okay, right? You don't need to be able to synthesize all of them. So that might be a good strategy to just ballpark it at the beginning. But indeed, it's very complex knowledge to incorporate directly. That's my feeling.
Yep.
Yeah.
Question.
Yes, I also
right
have
there.
a question. Can you hear me from
A
the
bit.
back? Okay, okay. So you mentioned this very large chemical space, so I'm curious whether it is desirable to explore the entire space, and if so, whether or what are the largest hurdles in exploring this very large chemical space, whether it's computing power, or whether you say out of this large chemical space, you only look at a fraction anyways.
Yeah, so the thing is that we don't know what's in there until we go there, right? So yeah, I guess we could consider that most of the molecules in there won't more likely be drugs, right? So most of it is not useless, is useless at least for drug discovery. It might be more useful for other tasks. But then of course, how do you know? You only have to know, you can only know when you get there. Yeah, and the hurdles are really, well, first of all, about how do we actually explore that? How do we enumerate all these molecules, and how do we navigate all of these molecules? And deep learning is really giving us a hand there. Before it was super complex. You had to define building blocks and assembly rules and see whether they would make sense. And you would see that even the best efforts in the field would still lead to a very small portion of the molecules we actually know already. So I would say in this sense, what deep learning enables us to do is to explore in a smarter way. Just say, okay, I don't want to explore it all, but for my task, I want to position myself here. So I think that's the sensible approach to adopt here. But yeah, the hurdles are still the diversity. How do we keep the property we want, but jump away from scaffolds we know and chemotypes we know, substructures we know? That's really still an open question. I'm not sure we will ever actually solve it, but let's see.
Yeah, speaking of diversity, I was thinking about evolutionary algorithms. I'm not sure whether you're familiar with them, but they are usually known for, or they are used in fuzzing in computing science, for generating some quite unique outputs. So I thought maybe that would be...
Yeah, that's a good idea, and people have done it already. So basically you can, for example, evolve smile strings or evolve other representations of molecules. But one tricky element of this is how do you, so usually you have a fitness function that steers your evolution like you would have in nature. But basically what fitness function do you use? Because if it's a predicted property, then you end up with the problems we saw, that this is just a model of the reality, and these methods are very good at exploiting, let's say, taking shortcuts to just maximize something without actually leading you where you want. So this is a very good idea, but it's still not perfect. It has its own caveats, and yeah.
Thank you for the explanation.
Question over there.
Wait, the microphone is coming.
Yeah, does this work? Okay, perfect. Sorry. I have a bit technical question about the data, right? So you said you need big data to analyze the molecules and generate new ones. And I'm thinking like, because there is the graph neural networks that can make use of equivariances and the invariances. Have you ever tried something like that?
I didn't. Some of my previous co-workers worked quite a bit with that. Yeah, and I mean, let's say this equivariance is mostly relevant when you're working with 3D structures, which is not something we're doing. And for sure, it's very cool and very interesting that you can have this equivariance. But one of the limitations, sorry, I always speak about limitations to just show how complex it is, that one of the struggles we have is that we don't know the 3D structure that a molecule has when it binds to a protein. So you could try to compute that to get as close as possible. But so far, in my experience, is that this introduces so much noise because it's still an estimate, it's not necessarily close to the truth, that sometimes 2D methods work better. And it's quite surprising if you think about it. But I do see that more and more people are starting to work in this domain, and I think we will get somewhere. Not me, I don't work with this, but it's very cool indeed as a direction. Yeah.
Okay, thank you.
There's a question from Hannah Raab in the chat. She asks, if you have to guess, when do you think an AI-generated drug will become available to patients?
Oh, okay, that's difficult. If I had to guess, well, I think that unless we change the standard pipeline, the molecule will still have to go through that. So animal tests and then clinical trials. So I would say 10 years, something like that.
I would probably think it's faster than that, because I think Exscientia and Benevolent AI,
Yeah.
these are large companies working together with pharma companies. I think they have drugs already in advance, sort of, at least in trials, I think. So
Yeah.
it might not take 10 years.
Yeah, maybe you're right, but I want to be cautious here and not make any overstatement. Let's say from 5 to 10.
Okay, we sort of
as an upper limit.
We just sort of guess. We don't know what.
Yeah, we can take a bet if you want, and then I call you in 5 years.
Yes, of course. But then I have forgotten. But now we have it on tape.
Yeah, exactly. There's proof.
Yes. Other questions? Yes, Emma.
I was wondering if there are types of drugs that are more suitable to be predicted by these models, or are there sort of types of drugs that are always going to be difficult?
That's a very good question. So what you can assume usually with these deep learning models is that, or in general with machine learning, is that because the model learns from data, this model will not be very good at going too far from the distribution of this data. So imagine we went there and had this model that would link proteins to ligands and use all of the data we have. Still, if you have a protein that is too different from all of the other proteins you trained it on, or need a drug that is too different from everything else, then you will struggle by definition, or I mean, in most of the cases.
If we already have many drugs for a certain disease, then we have more chance of finding.
Yeah, exactly. The closer you can be to what we already know, the better in general. As a rule of thumb, the easier it would be.
Maybe time for a last question. I have one that we haven't asked for a while, but I thought in this case it's maybe a good one. What would you be doing if you weren't a scientist? I guess you will become an environmentalist, but maybe maybe that's a question for you.
I think I would want to be an artist.
An
if
artist?
I could. Yeah.
Very good.
Yeah. Still don't know which type of art, but like painting maybe, or something like that.
You're not worried that DALL-E will sort of supersede your efforts very quickly?
Maybe I could use DALL-E and be a machine learning artist.
Machine learning
That could be a good
Very
idea.
Good. Very good, thank you. Francesca, thank you so much for entertaining us tonight and teaching us a lot about AI and drug discovery. It was a very insightful lecture. I thank you all for questions. I thank the people online for watching. And I would like to thank the team behind me also for making everything run very smoothly. That's it for tonight. I hope we see you back in a month's time when we have Theo Leino. He was already, well, he's still on the slide from IBM Zurich. He will be here in a month's time, 8 p.m. And we will hear more what IBM Zurich is doing in the field of chemistry and AI. That's all we have for tonight. Let's thank Francesca, and then the bar is open for those of you who are here, not the people online. Thank you.
Thank you.
Thank you.
Thank you.